Hello, everyone. Our next keynote speaker is Dr. Isabel Dorado Liñan from Spain. First time I met Isabel more than 20 years ago, yes, in Switzerland when we both were there, and she was a master's student at the University of Alicante then. Now she's associate professor at the University of Alicante, not in Alicante, actually I got lost here. She's associate professor teaching climatology in the Department of Natural Systems and Resources at the Universidad Politécnica de Madrid, actually. Yes, and over the years she has made significant contributions in the fields of forest ecology, paleoclimatology, and climate change research in general. Her research primarily focuses on understanding the connections and feedback mechanisms between forest ecosystems and the climate system in the context of anthropogenically induced climate change. For that, she uses a combination of different approaches and data sources, but tree ring data are her main tool for understanding forest-climate interactions. She aims to develop approaches for regional interpretations of climate impacts on tree growth that can be directly compared to the outputs of regional and global models. That's something very particular for her research. And her overarching research goal is to close the gap between empirical and modeling communities. And so I guess she is, with her research background, qualifies quite well for us here as a keynote speaker in actually presenting session number 4. And I would like to welcome her here, and please start your presentation.
Thanks to everyone for being here. Thanks to the conveners of the session 4 for inviting me to give this talk. I think it's the first time I'm in a conference with critical zone scientists. So my background, as Ingo mentioned, is a little bit different. I'm a forest ecologist. So my aim here is to introduce you how tree rings may help or be of some help in the critical zone research. So I will start with some basic concepts. I know that vegetation was one of the last components that have been considered in the critical zone research. And the reason why it's so important is because it's the living mechanism between the soil and the atmosphere. And this is important for mainly 2 fluxes. One of them is the water flux. As you all know, trees are uptaking water through the roots and they release via the stomata, which are little pores on the leaves, through the atmosphere, and then the carbon follows a reverse path. They uptake carbon through this stomata, and then they store it in the biomass, basically in wood, but also other compartments on the tree. In this talk, I will focus on trees, not on the all vegetation types, because trees have a larger implication for the critical zone processes, since they are the main carbon stock on Earth, are responsible for removing 1/3 of the CO2 emissions annually. And I will focus on that, but most of the things I'm going to explain could be applied to other type of vegetation. And also to mention that there are other processes where vegetations are involved, which is, for instance, the nutrient cycling, also the energy balance, and of course, they are strongly implicated in keeping biodiversity, but I won't enter into these topics in this talk. How the trees regulate those fluxes. So basically, the mechanism they have is the hydraulic strategy. The hydraulic strategy is how a tree is behaving under, particularly when there is some environmental constraints. So we can see this hydraulic strategy better when the trees are growing under a heat wave or they are suffering a drought episode. Historically, there have been defined 2 types of strategy that here I name saving strategy and business as usual strategy. The official names are isohydric and anisohydric strategy, and they're pretty contrasting. The first one, the business as usual, means that when the tree is under a heat wave or the drought stress, what they do is closing the stomata. Like this, they are saving water. They are preventing to lose water from evapotranspiration or from transpiration, but they are also preventing uptaking carbon. So they also reduce their growth. On the other side, we have the business as usual strategy, which is the anisohydric strategy, where trees just keeping functioning as normal, as nothing is happening. So stomata keeps open. They are able to continue growing, uptaking carbon and following with all their processes, but in the counterpart is the fact that they are losing out of water through transpiration. What is the best strategy? It depends. Usually it depends on how long is that dry spell or heat wave lasting. Now we know that those strategies are not that fixed. It's not only black and white. There is a lot of gray in between. And traditionally, those strategies has been assigned to specific species or plant functional types. So usually the pines, for instance, are described to have a saving strategy, whereas, for instance, oaks or beech are described to have a business as usual strategy or anisohydric strategy. This is important because this might imply of a difference of up to 30% or more transpiration or putting more water into the atmosphere. So under the same conditions, an anisohydric species, the one performing this business as usual strategy, will put 30 more water into the atmosphere compared to the saving strategy. This also means that it will uptake carbon because they keep the stomata open. And the carbon they will take is not 1-to-1. It will be something between 0 and 20. It depends on many other factors. But just to give you an idea, how will be the difference of the water you have in the atmosphere depending on the strategy they have. So now in the real world, how we track this, how we track the strategy that the trees are having. So here I just draft a little bit the observations that we can, sources of information or observation that we can have to track these processes on forests. So on the one hand, we have the remote indirect observations that now are pretty being used, and actually they have an uncomparable spatial resolution. There is nothing that is comparable to that. The limitation they have is that you still need the ground-based observations, the in situ observations, to validate that data. Within these in situ observations, we have, of course, the Tereno and OZCAR observatories and also other monitoring programs such as ICOS. Still, the time span they cover is pretty short. It's a few decades. So I think best case scenario, a Tereno site is covering 15 years or 17 years. And I think OZCAR shorter. In longer records, longer observational records, we have the National Forest Inventory data. This actually is not tracking physiological behavior, it's tracking basically biomass stock on forests, and does not have an annual resolution, is decadal. Measurements are taken every 10 years, or I think in France, the National Forest Inventory is every 5 years. And then we have tree rings. Tree rings, they can be as long as the tree lives. So this can stand centuries and even millennia. Just to give you a short overview, what is the data we are getting from an in situ observational plot. This is the Tereno Northeastern German Lowland Observatory that I helped to set up 15 years ago with Ingo. We were installing all these sensors, and there is a bunch of sensors on that side. For hydrology, there is also a meteo station, but I will just focus on the ones that are helping us to track processes on trees. One is the subflow. That very simple way is just tracking water fluxes on the tree. And then we have the dendrometers, which is this one here, is tracking how the tree is growing in thickness, which is also a measure how much carbon is storing into the wood every year. For the time period, this actually gives us an idea of how is the coupled carbon-water cycle in this forest. As I said before, these observational or these monitoring sites cover the last 15, 20 years best case scenario. So whenever we need longer records is when we need to go to the tree rings. And actually they give us analogs to the monitoring data. On the one hand, we have the simple thing, which is the tree ring width. I guess all you know, in temperate climates, a tree is growing every year, it's adding a new layer And you can see those layers clearly when you cut down the tree. Tropical climates is a different story, but in temperate climates there is no problem with that, or almost no problem. From there we can get a tree ring width, which is tracking how the tree is growing, similar to the dendrometers. And then we can track processes at the leaf level using stable isotopes on tree rings. So there are one more, but I will just focus on the stable carbon isotopes that is actually telling us how efficient the tree is using water to uptake carbon, how much water is losing while uptaking carbon. And then the stable oxygen isotopes that is basically explaining how that tree, whether it's more or less stressed by environmental conditions, how it's coping with that. With these variables, we have the coupled long-term carbon-water cycle of plants. And why I'm mentioning long-term so many times? Because this is a conceptual diagram that I did based on the data from the 6th IPCC report on the biochemical cycles, and is how it's going to be the evolution of the two main carbon sinks on Earth, which is the oceans in this side and the forest. The forest is the main one. And as you can see, it's obvious that the spread, the uncertainty on the future projections is 3 times larger in forests than in oceans. This is basically because biological processes are a lot more difficult to parameterize in a model than physical processes. And also these, the land surface models that are the one making those projections are based in short observational data. So they are quite skilled in reproducing short-term data, but they lose their skills as long as we move from this observational data. They have been identified the 2 main sources of this spread on the projections. One of them is the classification of vegetation in plant functional types. This is quite a coarse classification in which we group 3 species in big groups, and we assume that they set like a fixed parameter, and we assume that they behave the same way all the time. And those grouping are made based on biological determinism. So we assume that all conifers are one group, all broadleaves are another group. And the other problem is that these simplify growth processes. Actually, growth in these models, or at least in the one that I know, is implemented as a fraction from the GPP, from the gross primary production. From direct photosynthesis, there is a fraction that is what is considered growth. This is on the way of being solved, because I think this afternoon there is a talk where they are going to present a model, or part of the model that will be implemented in a process-based model is actually describing radial growth, real radial growth of trees. So what is the problem of this? That trees do not behave the same way as I will show in this talk. They adjust water and use throughout their lifespan depending on what are the environmental constraints. This is called acclimation, and that is not presented in the models. That's not represented in the models at all. On the other hand, we know that secondary growth, which is wood production, has different environmental drivers. It's not directly coupled to GPP, and that's why we will see at the end the projections of the land surface models do not match what we observe in terms of biomass production. The first question that comes to one's mind is how strong acclimation can be. This can really be so dramatic. To answer that question, first I need to explain what is the distribution range of a tree species. A tree can grow in a certain geographical area that usually is limited in the north, but conditions in the north or the south, depending if we are in the northern or the southern hemisphere, but it's limited in one side, but conditions that are too cold or too wet for that tree species. And then we have the trailing edge, when the conditions are too hot or too dry for that tree species. In this trailing edge is where we will see the strongest acclimation of the trees, because those conditions are pushing the tree to the limits of the physiological capacity. So we decided to see, we had a question, and we were wondering whether this classification in plant functional types was appropriate, because in the trailing edge of the species distribution, we observed that some species that theoretically were supposed to behave in like a business as usual strategy were applying an isohydric strategy under stress. So we selected 42 forests from 2 different PFT, conifers and broadleaf. And those are the main, those 5 species here are the main 3 species in Europe. And all of them reach here the southernmost limits of the distribution in Europe. And this is a map with the mean temperature for June, July, August. And you see that they are in the warmest parts of the gradient. We projected how these trees were growing under different climate scenarios. And I'm just showing here the most severe climate scenario, like the one that we all hope that is not going to happen, and how these trees will be growing, whether they will grow more or less. These maps represent the slope of the linear regression. Basically, darker brownish colors is telling you that the growth reduction is going to be stronger. So what we realized just by looking at it is that the forests that were located in the Eastern Mediterranean were showing larger growth reductions than the others. And that was regardless of the plant functional type. It was the same for conifers and for broadleaves. And yeah, our conclusion is that projected growth seems to be independent on the PFT. It seems to be constrained by the climate conditions. But we didn't have the physiological proof of that. So we decided to run this analysis, sampling more sites. Now in this map, we have 42 sites distributed in the Mediterranean for the same 3 species. We measure stable carbon isotopes, and we calculate intrinsic water use efficiencies. That is this measure that is telling you what is the carbon, how much water a tree is losing to gain carbon, okay? And it's also a measure in these environments of how much those forests are constrained by the harsh climate conditions. So we wanted to see how was the shear variability among all these chronologies. So first we ran a principal component analysis, and we see with the first principal component, this first EOF in the steel, we have stronger values on the eastern part. So this means that these eastern forests manage water in a similar way. They share more variability, these forests, compared to the other ones. If we look at the second principal component, we see that there is kind of a dipole. We have one sign of values here, a different one here. And we have found this other times in the Mediterranean, that we have these dipoles. And this means that they have different environmental drivers at that time. There is something that is exerting a difference between the Eastern and the Western Mediterranean. The only thing that can do that, in fact, is climate. Okay, so if we compare it with, for instance, the same principal component analysis, but for summer precipitation, we observe that the first EOF also has a trend similar to the one we observed here, and the second principal component shows a dipole. This is actually the Western Mediterranean oscillation that sometimes induces a drought here, where there is no drought in the western part, and the other way around. So the conclusion of this is that the synchronization of the water use in this forest is driven by climate. So that's why they look more similar when they're in the same area. And actually, it's also telling us that climate is overriding that theoretical biological determinism, that this classification of isohydric and anisohydric based on their biology is maybe not that correct, as is not correct the classification in plant functional types. Then, as I said before, trees can live centuries and can live millennia. So it will be so interesting to see how flexible is that acclimation in the long run. So here I'm going to show a case is a chronology from the Pyrenees that I developed during my PhD, and that covers roughly 700 years. And for showing how this was evolving, I will use this framework that is showing in one of the axes how the tree is growing, whether the growth is higher or lower, how is the value of the stable oxygen isotope that is telling you whether the stomata are more open or closed, and what is the discrimination, which is also telling us how much carbon they are gaining depending on whether stomata are open or closed. And as you see, there are 4 colors on the floor, which actually defines the main physiological framework that those trees could go through their lifespan. So those are the 4 scenarios. Here we have a drought condition that the trees need to close stomata and save water. Here we will have the reverse. We have open stomata and it's exchanging water and carbon because there are no water limitations. And then we have those 2 scenarios that particularly this one is a little bit more complex. I will explain later. So how was the trajectory of this chronology throughout the 700 years? That's it. That's how that tree was behaving. So here, I mean, this is in a smooth series. We don't see the internal variability because we won't see anything. But what we can see is the 1300, they were in this part of the let's say, physiological framework. So here, something was limiting growth. It does not seem that was water. It's a case where water and carbon is decoupled. But this could also be because the rain origin was different. So the isotopic signature of the rain, of the water, was different. Then, at the beginning of the Little Ice Age, which was a colder and bright period, they enter into this area. They have water, they have light, and they reach the highest growth during that period in the 1700. From that 1700, they start decreasing in growth, and they move through this region, where they still have enough water, but something is limiting the assimilation of carbon. That could be the availability of CO2, but since the Industrial Revolution already happened, I doubt that that was CO2 limitations, so it could be nutrient limitations as well. And we don't see it because this series is smooth, but the most recent years are already falling here in the saving strategy. Now it's too dry for them, so they need to close stomata and change the strategy again. All this is just to show you that a forest in 700 years went through all possible scenarios in the physiological framework. This is how flexible acclimation can be. And then, last, I would like all this physiological acclimation and so on has a lot to be with the carbon that the trees have taken and stored. And as we know now, there is an increasing pressure to track forest emissions and removals because of the incredible high concentration of CO2 that we have in the atmosphere. So forests are the main carbon sink, but we still don't know how much carbon they were storing, or historically how heat waves or droughts were actually reducing their capacity to store carbon. So in terms of forest biomass, like stocks of forest biomass per unit area, we have this national forest inventory data that, depending on the country, go back to the 50s or even to the beginning of the 20th century in some countries. The problem with this data is that it's decadal. We have values every 10 years or every 5th year, depending on the country, but we don't have annual resolution. So the question is how we can interpolate this so we know how these have been varying annually. And this is what we are trying to do. We are trying to use the sensitivity of tree rings to environmental variables to choose predictors and interpolate this data. This is what we call data fusion. We select climate variables based on their sensitivity to some tree ring data available that is on the plots of national forest inventories, and with that, we are able to annualize the biomass on these plots. So actually, we did a pilot study in the north of Spain in 240 beech forests that are highly sensitive to climate. And when we interpolate all these data in every site and we average it, what we got was this. Those are the anomalies of biomass storage respect to the mean for this period. Okay, we took this initial period just to attest. And what we can see is that the largest biomass reduction was in 1995 in this forest, and the largest biomass increase was in 2019. Before, that was not possible. We will only have what was the anomaly of the decadal variability, not the internal variability. And what is showing us that, for instance, if we plot the individual values for the 1985, we see that there was a reduction of around 20% of biomass production in those sites, but line surface models like ORCIDE are only reporting something about maximum 10% of it. So it's catching the signal of what was happening that year. There was a biomass reduction, but it's underestimating the weakening of that carbon sink. And well, just the last slide, what I think is the future's perspective for the biological process in the research in biological process in the critical zone will be to bridge scales and processes by integrating ring widths with subflows, because what we see with true rings are patterns, but we need to understand what is the mechanism that is behind, and that will be with dendrometers and subflows. Then we already provide long-term physiological and growth constraints that could help to include or to incorporate these acclimation processes into the models, or at least to rethink how to organize the plant functional types so it matches this capacity of the trees. And then, of course, moving beyond benchmarking, we can use this data for data assimilation and hopefully reduce uncertainty in the projections of the land and carbon and water cycles. And yes, that's all. Thanks for your attention.